my last class, I discussed about the cell as a unit of life. So we are just continuing this particular portion. So today I shall start cell organelles. Cell organelles, so many organelles are there: plastid, nucleus, mitochondria, endoplasmic reticulum, Golgi complex, ribosome. So many are there. So today I shall start first with plastids and histides. So what is plastids? What are plastids actually? Plastids are pigmented or non-pigmented double membrane bound cytoplasmic organelles concerned with certain special functions of the plant. So that plastid, the term plastid was first given by Ernst Haeckel in 1866. So plastids are of three types. One is called leucoplast, one is called chromoplast, and one is called chloroplast. Leucoplast means colorless plastids, having no color or rather white in color. And these leucoplast we get in those parts of the plants which are generally not exposed to sunlight. We find them in such parts of the plants which are generally not exposed to light, say rhizome, tuber. These are the things. Rhizome means ginger, tuber means potato. So we find them which are not exposed to sunlight generally. So there are three types of leucoplast or colorless plastid. One is called amyloplast, 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 then elioplast, and another one is proteinoplast. Another one is proteinoplast, or alluroplast. So amyloplast, they are the plastids which, or type of leucoplast we can say, which can store starch. We find them in rice, wheat, maize, like that. Elioplast, which stores oils and fats. In some oil seeds you may get them. And proteinoplast or alluroplast, which stores protein. So what are the functions of leucoplast? If I say that leucoplast serves three main functions. One is storage of starch, one is storage of oils and fats, and one is storage of proteins. Now next one is chromoplast. Chromoplast means colored plastid. Colored other than green and white. Other than green and white. And they are generally yellowish or reddish in color because of the presence of carotenoid pigments. And they may be of other colors also, means except green and white, whatever colors are there in the leaves or in the fruits or in the flowers, this is due to chromoplast. And these chromoplast we generally get them in flowers, we get them in some leaves, and we get them in fruits which are colored. Flowers are also colored except white flowers. And then chromoplast serves two main functions. One is formation of color in flower and fruits, and another one, as it is colored, it attracts insect and other pollinating agents, and as well as it also helps in dispersal of fruits and seeds. I repeat, it serves two main important functions. One is formation of colors in flowers, fruits, and some leaves, and another important point is that it helps in the process of pollination by some insects or other agencies as it is colored, and also helps in dispersal of seeds and fruits. Now the most important one, that is chloroplast. Most important one, that is chloroplast. So chloroplast These are green colored plastid. Green colored plastid. Green colored plastid found in leaves and green stem. Chloroplast may be of different shapes. In some algae we find cup-shaped chloroplast like this, and in some algae we find ribbon-shaped chloroplast like Spirogyra, like this, and in some lower algae may be plate-like. But in higher planes, chloroplasts are generally biconvex or planoconvex in shape. This is the biconvex or planoconvex in shape. So, in leaves of higher plants, we find chloroplasts which are biconvex or planoconvex in shape. So, if we see the internal structure or ultrastructure of chloroplast, then we see chloroplast is bounded by two membranes, the outer one and the inner one. And inside chloroplast we get two things. One is called stroma or matrix, and another one is called grana. Now, stroma or matrix means inside the membrane of the chloroplast some fluid filled substances or matrix are there, that is called stroma or matrix. And what is their function? First of all, they carry out dark reaction of photosynthesis. Photosynthesis, we know of two types. One is called light reaction, one is called dark reaction, that you will get this here. And inside the stroma or matrix some granular bodies are there which are called grana. I am showing it here. The total structure, these are called grana. And individual one, if we talk about this one, these are called granum. And inside the grana, some coin-like structures are there. Disc-shaped coin-like structures are there, which you call thylakoid. Thylakoid, thylakoid. And I repeat, these are inside the stroma or matrix, these granular bodies are present which are called grana. An individual set of cells we call granum. And in each granum you see some disc-shaped structures are there. Which are called, or coin-shaped, disc-like coin-shaped structures are there which are called thylakoid. Now you see different in a chloroplast, how many grana are there? Maybe 40 to 60 grana or even more. And each granum, individual one we call, total one called granum, and individual segment in the granum is called thylakoid. I told you. Each granum, this is one granum, this is another granum, this is another. They are connected by a tube-like structure called frets or lamellae. These tube-like structures are called frets or lamellae. These grana are meant for light reaction of photosynthesis. Now these grana, I said that they are made up of thylakoid. These are thylakoids. Individual segment is called thylakoid. Now this thylakoid contains special type of photosynthetic unit called quantosomes. Contains special type of photosynthetic units called quantosomes, which contains different types of enzymes, proteins and etc., and carries out the light reaction of photosynthesis. Now, what is the function of chloroplast? The main function of chloroplast is to carry out the process of photosynthesis. Now, next thing, different function, difference of chloroplast, chromoplast and leucoplast. Just little bit I am telling you. Leucoplast, then chloroplast, then chromoplast. Now leucoplast, as I said, colorless plastid. Then chloroplast, I said green colored plastid. Chromoplast colored other than green and white, reddish yellow. Then the function of leucoplast is storage. Function of chloroplast is photosynthesis. Function of chromoplast, I said, formation of colors in flowers, fruits and leaves. Then leucoplast can change into other type of chloroplast, maybe other type of plast, that is maybe chloroplast or chromoplast. Chloroplast can also change into chromoplast, but chromoplast will never change. Now another important thing, we call chloroplast as semi-autonomous organelle. It may come in the exam. Chloroplast as semi-autonomous organelle. Semi-autonomous organelles. Why they are called semi-autonomous organelles? Because of some reasons. Say, if I say that chloroplast possesses its own DNA, and that DNA can replicate itself. That means DNA of chloroplast can produce another type of DNA. Then this one also, the DNA of chloroplast also undergoes replication, means forms different types of RNA. Then another important thing, chloroplast contains proteins, enzymes, and performs most of its own functions. Then each chloroplast can form from pre-existing chloroplast or from some precursor of chloroplast called proplastid. So these are the reasons. I repeat, first of all, it contains DNA which helps in synthesis. It contains DNA which can replicate itself, means from one DNA another DNA can be produced, and this DNA also undergoes the process of transcription, that is, it forms in different types of RNA. Then chloroplast contains different types of proteins, different types of enzymes and other substances that carry out different biochemical reactions. Then I said each chloroplast exists from pre-existing chloroplast, means it divides and forms from the pre-existing chloroplast, or it can be formed from some proplastid, means precursor. So these are the reasons we call chloroplast as semi-autonomous organelle. Now another important thing, as a short question you may get, chromatophores. Chromatophores. Chromatophores, what are chromatophores? Chromatophores are plastid-like coloring pigment. I repeat, plastid-like coloring pigment found in some bacteria and Organelles. Plastid-like coloring pigment, not exactly plastid-like coloring pigment found in bacteria and some Organelles. And now there are different types of chromatophores, you can say one is called rhodoplast. Rhodoplast found in plastid of red algae. Then pheoplast, plastid of brown algae. Rhodo means red algae, plastid found in red algae, and pheoplast means plastid found in brown algae. So this particular different types of plastid is over. Now we shall go to nucleus. I must start another one, nucleus. So nucleus, all of you know, a cell contains two main parts. One is called contains main parts called protoplasm, which consists of nucleus and cytoplasm. What is nucleus? Nucleus is the headquarters of all cellular activities. And nucleus was first of all discovered by Robert Brown. Robert Brown in plant cell. Robert Brown in plant cell in 1831. In 1831, nucleus is found in all types of eukaryotic cells except mature mammalian RBC and sieve tube in plants. Then in prokaryotic cell, nucleus do not have any cover, and that nucleus is called nucleoid. Nucleoid means nucleus without nuclear membrane. In prokaryotic cell, nucleus do not have any cover, and it is called nucleoid, means nucleus without cover. And in a cell, the number of nucleus varies. Mostly it contains one nucleus. If one nucleus is there, it is called uninucleate. Uninucleate or mononucleate. Mononucleate. In some protozoa, two nucleus is present. It is called binucleate. Binucleate or dinucleate. And in some cells, more than two nucleus is present. That is called polynucleate. Polynucleate or multinucleate. Multinucleate. This is found in bone marrow cell, then striated muscle of human being and other animals. So I repeat, nucleus may be of three types: uninucleate or mononucleate containing single nucleus, as in most of the cell, binucleate or dinucleate in some protozoa, and polynucleate or multinucleate in our bone marrow, striated muscles, etc. Now this nucleus. Now let us go to the ultrastructure of nucleus. Now let us discuss about ultrastructure of nucleus or electron microscopy structure of nucleus. Nucleus consists of 5 main parts: nuclear membrane, nuclear pore, nucleoplasm, nucleolus, and chromatin reticulum. This membrane, which is a double membrane structure, is called nuclear membrane. Nuclear membrane that separates nucleoplasm from cytoplasm, and this nuclear membrane is not continuous. After some interval, some gaps are there which are called nuclear pore, and the space between nuclear membrane, this space between the two membranes, is called perinuclear space. And on the outer side of the nuclear membrane, some cell organelles are attached, such as endoplasmic reticulum, ribosome membrane, maybe mitochondria also. Then next structure called nuclear pore. Nuclear pore means gaps over the nuclear membrane is called nuclear pore. Through this nuclear pore, nucleoplasm can move to the cytoplasm or for performing some functions. These nuclear pore are covered by a ring-like structures, like that ring-like structures. Suppose these are they are covered by some ring-like structure called annulus. Annulus, which just maintains the movement of the nucleoplasm from the nucleus or cytoplasmic part. Then apart from nucleoplasm, then let me talk about nucleoplasm. Nucleus is filled up with some granular transparent substance which contains different types of enzymes, water, minerals, that we call nucleoplasm, which carries out maximum function of the nucleus. And within this nucleoplasm, some thread-like structures are there called chromatin reticulum, and one spherical structure called nucleolus. So now let us talk about nucleolus. Nucleolus, a spherical structure which is membraneless, having no membrane, and it may be found in three forms, such as granular, then fibrillar, and amorphous form. And this nucleolus acts as the nucleolar organizer, means it helps in formation of chromosome and other, also performs different functions. The next one, thread-like structures that we call chromatin reticulum. Chromatin reticulum, you can see in your diagram, two types of chromatin reticulum is there. One is called euchromatin, another one is called heterochromatin. Euchromatin means which are less coiled. Heterochromatin, can you see the black dark portion? These are called heterochromatin. If we go through the differentiation of euchromatin and heterochromatin, we can say euchromatin is less coiled and less condensed. Heterochromatin is more condensed and more coiled. Then euchromatin do not take strain, means color. Heterochromatin takes stain. Then euchromatin is rich in DNA and is genetically active, but heterochromatin contains less amount of DNA and is genetically inert. And in case of euchromatin, crossing over frequency is more, but in heterochromatin, crossing over frequency is less. Now let us discuss about difference between nucleus and nucleolus. It may also come in the exam. Nucleus means part of protoplasm. Nucleolus means part of nucleus. Nucleus is bounded by nuclear membrane. Can you see here? These are the membrane, double membrane envelope. But nucleolus lack any membrane. Then another important thing, nuclear membrane of nucleus may remain attached to some cell organelles like ribosome, then endoplasmic reticulum, mitochondria, but nucleolus has no such attachment. Then nucleus is less condensed, but nucleolus is more condensed. Once again I am saying, nucleus is a part of protoplasm, nucleolus is part of nucleus. Nucleolus is having membrane or nuclear membrane, you call it. Nucleolus do not have any membrane. Nucleus is bigger in size, nucleolus is smaller in size, and nucleus is not so condensed, but nucleolus is so condensed. So with this we will finish today, and the next day we will discuss about chromosome and its types.